Hello everyone. Today we are going to start chapter 21 of level 2, Principles of Genetics, Heredity and Variation, Important Terms in Genetics, Incomplete Dominance, Linkage and Crossing Over, Sex Determination, Chromosomal Disorders, and Human Genome. So, Principles of Genetics. It is common observation that seeds of mango trees grows into mango plant. Dogs give birth to puppies and not into the young ones of any other animal. Human gives birth to human beings. The tendency of offspring to inherit parental characters is termed as heredity, and the science of heredity is called genetics. Genetics also seeks to answer questions like why two offsprings of same parent look different, why are some people dark and other light skinned, etc. In other words, why is there variation among individuals of the same kind? This lesson deals with heredity and variation. It also includes a section on hereditary disorders and gives an idea of the human. So after completing this lesson, you will be able to explain the terms heredity and variation, describe Mendel's experiment on garden pea plant and the principles derived, define the term hybridization, alleles, trait, dominance, recessive, homozygous, heterozygous, genotype, phenotype. Explain the term incomplete dominance with the example of flower color in 4 o'clock plant, Mirabilis jalapa. Define the term lethal genes and pleiotropic genes. Explain quantitative or polygenic inheritance supported by the examples of kernel color of wheat and skin color of humans. Differentiate between monogenic and polygenic inheritance. Explain the chromosome theory of heredity. Define and give example of linkage, crossing over, and criss-cross inheritance. Justify mitochondrial inheritance as a case of maternal inheritance. Describe the human karyotype. Differentiate between sex chromosomes and autosomes. List and describe the causes and symptoms of some common genetic disorders: color blindness, hemophilia, Down syndrome, Turner syndrome, Klinefelter syndrome. Describe the inheritance of RS factor and explain its significance during pregnancy. Explain ABU blood group. Explain the diagnostic technique of aminosynthesis and give its significance. Give a brief idea of human genome, heredity and variation. Whenever an infant is born in a family, the relatives begin to wonder about the resemblance of the infant's eyes, facial features, complexion, color of hair with those of the parent, sibling and grandparents. The source of such resemblances and differences are in the genes that are passed down from parents to children and so on generation after generation. This inheritance of gene is called heredity and the study of heredity is called genetics. New individuals develop according to the genes inherited from their parents. The transmission of characters from one generation to the next, that is from parents to offspring, is known as heredity. It is further observed that siblings from same parents are unique and differ from each other except the identical twins. Such differences are called variations. Variation means differences between parents and offspring, or between offspring of same parent, or between members of the. Variation in a population is very important. It has survival value for the population. This is because if the environment changes, some individuals may be able to adapt to new situations and save the population from dying out. Variation arises due to mutation or sudden change in the genes. Variation also arises because genes get shifted and exchanged during meiosis at formation of gametes, giving rise to new gene combinations. Just recall from lesson 8 on cell and cell division and about chiasmata formation, and lesson number 20 on reproduction in animals for gamete formation and fertilization. At fertilization, there is random mixing of parental chromosomes with different gene combinations. Such a source of variation, which is most common, is called recombination. Variation arises because of mutation and genetics is the branch of science that deals with the study of heredity and genes. Pythagoras, Aristotle, Hippocrates, Epicurus, and others have contributed to the field since the classical period. Modern genetics was pioneered by Gregor John Mendel. His work on pea plant Pisum sativum was published in 1866. This established the theory of Mendelian inheritance. Inheritance is the process by which characters are transferred or passed out on from parents to progeny. Heredity is based on inheritance. Variation is the degree to which progeny differ from parents. Variation can be in the sectors of morphology, physiology, cytology, and behavioristic. You can see these all scientists who worked after Mendel: Sutton and Boveri, Muller, Emmerling, MacLeod. So please study this whole chart. Mendel has a great interest in plant hybridization from the start because his father was interested in the same field. Mendel conducted his historic experiment with garden pea in the monastery garden for about 9 years and published the result in a less known journal in 1865. Plant selected by Mendel is Pisum sativum. Mendel used several contrasting characters for garden peas, which are represented below. You can see in this chart: seed color, pod shape, pod color, round, and first one is seed shape, flower color, flower location, and plant. So Mendel selected garden pea as material in his experiment due to the following advantages. Pea plants are easy to grow and interbreed. Peas reproduce and grow to maturity in a season. Several generations can be produced with a short period due to short life cycle. The flowers are bisexual, containing both female and male parts. They are self-fertilizing in nature. Several contrasting characters are present in pea plants. It is easy to hybridize because pollen from one plant can be introduced into the stigma of another plant by removing anthers. This process is also called emasculation. Mendel's law. Mendel first performed a monohybrid cross in which he crossed tall and dwarf pea plant and collected all the seeds obtained from this cross. He grew all the seeds to produce plants of the first hybrid generation. This is called F1 generation. He observed that all the plants are tall. Mendel self-pollinated the F1 plants and discovered that in the F2 generation, some plants are also dwarf. The proportion of plant is 1/4 and that of tall plant is 3/4. You can see this checkerboard. Tall and dwarf, capital TT crossed with small tt, then selfing them, and he got 3:1 ratio, 75% tall and 25% dwarf. Law of dominance. It states that characters are controlled by discrete unit. These are called factors. Factors always occur in pairs. In a dissimilar pair of factor, one member of the pair dominate the other member, law of segregation. The allele do not blend and both the characters are recovered during gamete formation as in F2 generation. Law of independent assortment. The law states that when two pair of traits are combined in a hybrid, segregation of one pair of characters is independent of the other pair of characters. Two new combinations, round green and wrinkled yellow, are formed in the dihybrid cross due to an independent assortment of traits for seed shape, round wrinkled, and seed color and green. You can see this. This is called Punnett square. You will find three triangles in this diagram, which represent ratio 9 is to 3 is to 3 is to 1, and this is phenotype. So points to remember: dominance, when a factor expresses itself in the presence or absence of its dominant factor, called dominance. Recessive, it can only express itself in the absence of or its recessive factor, allele. Genes that code for a pair of contrasting traits are alleles. Some exceptions of Mendel's law. Mendel's law too had some exceptions. Here is the list. Incomplete dominance. It is a post-Mendelian discovery. It is the phenomenon of neither of the two alleles being dominant, so that expression in the hybrid is a fine mixture or intermediate between the expression of two alleles. In Mirabilis jalapa, there are two types of pure breeding plants, white flowered and red flowered. On crossing the two F1 plants, it possesses pink flowers. On selfing them, the F2 generation has one red, two pink, and one white flower. The pink flower are due to incomplete. Just watch it. This is the example of incomplete dominance in Mirabilis jalapa. Codominance. It is the phenomenon of two alleles lacking dominance recessive relationship and both expressing themselves in the organism. Human being ABU blood group are controlled by gene I. The gene comprises three alleles, IA, IB, and small I. Any individual contains any two of three alleles, IA, IB, are dominant over small I. The plasma membrane of RBC has sugar polymers that protrude from its surface, and the kind of sugar is controlled by the gene. When IA and IB are present together, both express their types of sugar due to codominance. Multiple alleles. They are multiple forms of a gene that occur on the same gene locus, distributed in different organisms in the gene pool, with an organism carrying only two alleles and the gamete containing only one allele. For example, ABO blood. So now we started going deeply Mendel's experiment on the garden pea and principles of inheritance. Sir Gregor John Mendel in 1822 to 1884, he was an Austrian monk who used garden pea for his experiment, and he published his result in 1865. His work, however, was rediscovered in 1900, long after Mendel's death, by three scientists, Sharmak, Carens, and De Vries. But since Mendel was the first to suggest principles underlying inheritance, he is regarded as the founder of genetics. Wait a minute. So here you can see introduction. So Gregor John Mendel conducted experiment to answer one of the fundamental question of heredity, that what are the basic pattern in the transmission of traits. So in this recreation of Mendel's historic experiment, you will have the opportunity to predict what is the outcome. In this first example, the plant being mated after differ in just one trait. One plant is homozygous for the dominant purple flower allele of a gene. The other is homozygous for a recessive white flower allele. To show the mating, we arrange the parent's plant along two sides so that you can, yes. Note that each parent produces a single type of gamete. The genotype of each gamete produced by one parent is brought down into each of the square below it. Likewise, the genotype of each gamete produced by the other parent and moved to each square to the right. So it held, this is called known as Punnett square. So exercise 1, using the information provided predict the phenotype of the offspring. So drag the correct phenotype to each square in the grid. So notice that all of these offspring have the same flower phenotype as the purple flower plant in the parental generation. So this is called F1 generation. The white phenotype disappears. If we mate these heterozygous plants to each other or allow them to self-pollinate, then what would happen? Could you expect? So from the choices given, select the current gamete to place on this. Number 3, using the information provided predict the genotype of the offspring. Try to fill this Punnett square. So using the information provided predict phenotype of the offspring. Drag the correct phenotype to each square. So inheritance of one trait. Conclusion. The first series of crosses recap the result of mating, which the parent plant differ in a single trait. So the parent plant are homozygous for the dominant and recessive for the alleles. You can see this, and got ratio is 3:1. This is the phenotype. And recessive white flower disappeared in the F1 generation. So the two series of crosses shown here illustrate a mating in P, round and wrinkled. Second is yellow and green. In each series 3:1 ratio he got. If the two heterozygous F1 plant were crossed, what is the genotype and phenotype? Can you guess? Just imagine. So please look at. These are homozygous parent plants, differ in both the R and the Y genes. Look here. The round yellow pea has dominant for a round phenotype and dominant Y for a yellow phenotype. The wrinkled green pea has recessive R. So from the choices given, select the correct gamete to place on the two sides. Try yourself. Pause here and try yourself. Clear? In our cross, notice that all the F1 individuals show dominant yellow and round phenotype. It means capital I, R and capital Y is present. So next, just notice after selfing, what is the phenotype and genotype? So please being dragging correct. These are the choices. And F2 generation just cross and you will get three triangles here. 9:3:3:1 ratio. 9 are round and yellow, 3 are round and green, and 3 are wrinkled and yellow. 1 is pure wrinkled and green. It means pure homozygous. According to Mendel's principle of independent assortment, the genes behave independently. So try to make this Punnett square by yourself. So although our cross involved a difference in two traits, we can examine each trait individually, like only single character, and you can choose both characters together and just tell about what is the genotype, because you know Phenotype is 9331. Clear? I hope you can understand. So try to do this one. Mendel's experiments. Mendel designed his experiment such that a pure tall variety of pea plant could be crossed to a pure dwarf variety. The anthers from flowers of tall plants were removed and their stigmas dusted with pollen from flower of dwarf plants. The reverse experiment was also carried out. In the following, spring seeds from the new plants were collected and sown. He found that all the plants of this generation, called first filial generation or F1 generation, these are tall plants. He allowed them to self-pollinate. Again he collected the seeds. The following year, after the seeds had been sown, he found that 3/4 of these plants were tall and the rest are dwarf. He repeated the experiment several times and found that the ratio of tall to dwarf plant was 3:1. In this way, he tried to cross pea plant with 7 such contrasting characters. These were red flowered and white flowered, axillary flowered and terminal, yellow seeded, green seeded, round and wrinkled, green pod versus yellow pod, plant with inflated pod versus constricted pod, pure tall plant, pure dwarf. So plants with such contrasting characters exist in varieties that are self-pollinating, so that generation after generation they express only one type. You can see in the next figure. Just watch it. So crosses considering the inheritance of one feature only are called monohybrid crosses. Mendel also tried crosses involving two contrasting features such as tall and red flowered with dwarf and white flowered plant. Such a cross is termed as dihybrid cross. Mendel's principle of inheritance. Based on the result of his experiment, Mendel postulated the following laws of heredity. Law of segregation or purity of gametes. At formation of gametes, the two chromosomes of each pair separate into two different cells which form the gametes. This is a universal law, and always during gamete formation in all sexually reproducing organisms, the two factors of a pair pass into different gametes. Each gamete receives one member of a pair of factor and the gametes are pure. Look here, seed shape, seed color, flower color, inflated, constricted, green, yellow, terminal, etc. So law of dominance, inheritance of many features like eye color, flower color, seed shape, etc. is controlled by one pair of genes. When the two genes are of the same kind, like brown color of eyes, red color of flower, the condition is termed homozygous. When a pair of chromosomes has the gene controlling the same feature in two different forms, red flower gene on the one chromosome and white flower gene on its pair, this is called homolog. So this condition is known as heterozygous. The second law of inheritance maintains that of two contrasting genes of a pair, the expression of one is dominant over the other. Thus, if both genes are for tallness, that is homozygous, or for dwarfness, so capital T and small t is these plants will be tall. Opposite of dominant gene is termed recessive gene. The recessive feature dwarfness of the plant shows up only when its genes is in the homozygous condition. The law of dominance was found to be true in both monohybrid and dihybrid crosses. In cases of the characteristics studied by Mendel in the garden pea, law of independent assortment, meaning whereby that in the inheritance of two features, each feature controlled by a pair of genes for the two different features are passed down into the offspring independently. The segregation of one pair of factor is independent of the segregation. So this is the cross between red tall and white dwarf. So results showing independent assortment in two pairs of genes. R stands for red flower color, small r for white flower color, capital T for tall gene, and small t for gene for dwarfness. So you would have noticed that the composition of genes or genotype controls the outside expression, which can we say that is the phenotype. The ratio of progeny in the crosses is therefore the phenotypic ratio. However, as more and more scientists began to devise genetic experiments, it became clear that Mendel's law do not hold true in all cases. We shall learn about the deviations from Mendel's law such as incomplete dominance, codominance, etc. Reasons for Mendel's success. His choice of material was good. He selected garden pea, which has a short life cycle, has self-pollinated bisexual flowers with closed corolla, so pollination can be easily controlled. Also, it is easy to cultivate pea plants, and a large number of pure line plants with several pairs of contrasting characters were available. His selection of traits, the 7 pairs of contrasting characters of pea plant considered by Mendel in his experiment, were responsible for the desired results that helped Mendel postulate law of inheritance. Mendel's technique, his technique for experimentation was excellent and was as follows. Homozygous pure line plants with contrasting characters were crossed. Self-pollination was prevented by removing stamens. Female plants were dusted with pollen grains from another plant with the opposite feature and were tied in a bag to prevent any further pollination. Seeds were collected and shown in time. He considered the inheritance of one character at a time. The results were analyzed statistically. He performed. So these are some important terms in genetics. Factor, the unit responsible for the inheritance and expression of a particular character, now replaced by the term gene. Gene, it is a particular segment of a DNA molecule which determines the inheritance and expression of a particular character. Alleles or allelomorphs, two or more alternative forms of a factor or a gene, are called alleles. For example, in pea plant, the gene for producing seed shape may occur in two alternative forms, round and wrinkled. Genes for round and wrinkled seeds are alleles of each other. Similarly, there are 3 alleles for gene controlling blood group in man, IA, IB and small I. Alleles occupy same locus on homologous chromosomes. Trait, this is the expressed character like color of flower, shape of seed, etc. Dominant trait, out of the two alternating forms, these are called allelomorphs of a trait. The one which expresses itself in a heterozygous organism in the F1 hybrid is called the dominant trait and the phenomenon is called dominance. For example, in an organism with capital T small t, capital T expresses itself and small t cannot, so capital T is the dominant allele. Recessive trait, out of the two alternating alleles for a trait, the one which is suppressed in the F1 hybrid is called trait. Phenotype, the outward appearance of an individual for any trait or traits is called phenotype. For example, seeds, round shape or wrinkled shape in the phenotype. Homozygous, an individual possessing identical alleles for a trait is called homozygous, or pure, for that trait, plant with capital R alleles is homozygous for the seeds. Heterozygous. An individual receiving dissimilar alleles for a trait is called heterozygous or impure. Parent generation. The parents used for the first cross represent the parent generation. F1. The progeny produced from a cross between two parents is called first filial or F1 generation. F2. The progeny resulting from self-hybridization or inbreeding of F1 individual is called second filial or F2 generation. Monohybrid cross. The cross between two parents' offspring in a single pair of contrasting character is called monohybrid cross, and the F1 offspring as the hybrid. The phenotype ratio of 3 dominant, 1 recessive obtained in the F2 generation from the monohybrid cross is called monohybrid phenotypic ratio. This is 3:1. Dihybrid cross. The cross between two parents in which two pairs of contrasting characters are studied simultaneously for the inheritance pattern is called dihybrid cross. The phenotypic ratio obtained in the F2 generation from a dihybrid cross is called dihybrid ratio phenotypic 3:1 in Mendelian crosses. Hybridization. Crossing organisms belonging to different species for getting favorable qualities in the offspring. Test cross. Crossing of the F1 progeny with a homozygous recessive parent. If F1 progeny is heterozygous, then test cross always yield the ratio of 1:1. Reciprocal cross is a cross in which the sex of the parents is reversed. That is, if in the first cross father was dwarf and mother tall, then in the reciprocal cross dwarf parent will be female and tall parent male. Deviation from Mendel's law, incomplete dominance. In the 4 o'clock plant Mirabilis jalapa or Antirrhinum, law of dominance is not followed. Thus, when a homozygous red flowered plant is crossed to a homozygous white flowered plant, all flowers in the F1 are pink. When F1 plants are self-pollinated, the phenotypic ratio is found to be 1:2:1. Parents, gametes, F1 generation. F2, you will find that the heterozygous plants have an intermediate color pink. You must have also noticed that the genotypic ratio 1:2:1. Multiple alleles and codominance, tall or dwarf, red or white, brown eyes or blue eyes. These are just two varieties or alleles of genes for plant height and flower color and eye color. Most genes, however, have more than two alleles or multiple alleles. An example of multiple allele is the gene for blood group. The 4 blood groups of humans are determined by a single gene. The allele IA for A group, IB for B group, both are dominant. Therefore, person with allele IA and IB has the blood group AB, as both the genes IA and IB are codominant. The gene small I, when homozygous, gives the blood group O. So, genotype and phenotype of blood group in given in this table. You can see and compare them and try to figure out. Lethal genes. Have you ever seen a yellow mouse? Probably not. The yellow coat color in mouse is due to the presence of the gene, which is also responsible for killing the mouse born with that gene. Such genes are termed lethal genes. Some lethal genes kill only in the homozygous condition and are recessive lethals. Dominant lethals cause death even when present in the heterozygous condition. Pleiotropy. While a gene may have multiple alleles and thus give multiple genotypes, one gene may control several genotypes. For example, the recessive gene for white eye in Drosophila, when present in the homozygous condition, affects several other features such as wing shape and shape of abdomen. Thus, a white-eyed Drosophila is also born with vestigial wings and abdomen. Polygenic or quantitative inheritance. When a trait is controlled by a single gene, it is termed monogenic inheritance. Many traits or features are controlled by a number of different genes. For example, the skin color of human and the kernel color of wheat results from the combined effect of several genes, none of which are singly dominant. Polygenes affecting a particular trait are found in many chromosomes. Each of these genes has equal contribution and cumulative the total effect. 3 to 4 genes contribute towards formation of the pigment in the skin of humans. So there is continuous variation in skin color from very fair to very dark. Such inheritance controlled by many genes is termed quantitative inheritance or polygenic inheritance. In polygenic, each dominant gene controls equally the intensity of the character. The effect of the dominant gene in cumulative, the intensity of character or trait depend upon the number of dominant genes. Next is you can see and you have to draw this table by yourself. Chromosomal theory of inheritance. It was observed by Sutton and Boveri in 1902 that maternal and paternal characters come together in the progeny, which is diploid or 2n and has chromosomes in pairs and later on segregate during the formation of gametes. The gametes have a single chromosome from each pair and is haploid. Chromosomes from two parents come together in the same zygote as a result of the fusion of two gametes and again separate out during the formation of gametes. You have already learnt that chromosomes are filamentous bodies present in the nucleus and seen only during cell division. Wait a minute. The above two observations proved that there is a remarkable similarity between the behavior of characters during inheritance and that of chromosomes during meiosis. This led Sutton and Boveri to propose chromosomal theory of inheritance, and its salient features are as follows. The somatic cells of an organism, which are derived by the repeated division of zygote, have two identical sets of chromosomes. They are diploid. Out of these, one set of chromosome is received from the mother. This is called maternal chromosome, and one set from the father. Two chromosomes of one type constitute a homologous pair. Humans have 23 pairs of chromosomes. The chromosomes of homologous pairs separate during meiosis at the time of gamete formation. The behavior of chromosome during meiosis indicate that Mendelian factors or genes are located linearly on the chromosome. With progress in molecular biology, it is now known that a chromosome is made of a molecule, DNA and segment of DNA genes. Linkage and crossing over. Batson and Pinnett performed a dihybrid cross with true breeding varieties of sweet pea, and instead of 9:3:3:1 ratio in F2 generation, they got the ratio is 7:1:1:7. So the parental forms were more than a new combinations of characters. It means the two genes chosen for the experiment do not follow the principle of independent assortment. Instead, they to be inherited together or are linked together. Thus, genes present on same chromosome tend to be inherited together and are said to be linked. This phenomenon is called linkage. A group of genes present on the same chromosome and tend to be inherited together forms a link. So in the above experiment, some recombinant types were also produced. How did that happen? They are produced by another phenomenon called crossing over. Crossing over is the physical exchange of parts of non-sister chromatids of chromosomes of homologous pair. Crossing over occurs during meiosis 1, prophase 1 at the time of gamete formation. The point where crossing over occurs is called chiasma. So linked genes get separated from each other by crossing over. Because of linkage and crossing over, a heterozygous individual can produce 4 types of gametes as shown in the next figure. The figure shows linked genes of the parent and recombinants due to crossing over. You can see here in this figure chromosome and sex determination. In some diploid organisms, specific chromosomes have a role in sex determination. Such chromosomes are called sex chromosomes, and the rest of the chromosome of a set are called autosomes. If sex chromosomes are morphologically similar in an individual, the individual is termed homogametic. Such individuals produce only one kind of gamete. For example, all eggs of the human female contain an X chromosome, so human female is termed homogametic. If sex chromosomes are morphologically dissimilar in an individual, this is termed heterogametic. Such individuals produce two types of gametes. For example, human male produces two kinds of sperms, X-bearing and Y-bearing sperms. When the human egg is fertilized by an X-bearing sperm, a boy is born. Whether the unborn will be a male or female is purely a matter of chance, and no parent can be blamed for the sex of the progeny. You can see chromosomes, number of sex chromosomes and auto. So crisscross inheritance. We already know that genes are located on chromosomes. The genes which are located on chromosomes are called sex-linked genes. These genes show crisscross inheritance as shown in next figure. When a male has a definite sex-linked gene located on X chromosome during reproduction, he transmits the defective X chromosome to his daughter only. The female who has this gene transmits it to her sons and daughter, both in equal probability. So the male passes on his recessive sex-linked trait to 50% of his grandsons through his daughter. The sex-linked trait, being recessive, is not expressed in female but is expressed in male. Therefore, males suffer from genetic defect, while females are only carrier of these defective genes. Females express the trait if they are homozygous for the recessive gene, father and mother, hemophilic and normal. This type of inheritance of recessive sex-linked character from father to daughter and then from the daughter to her son is known as crisscross inheritance or sex-linked or X-linked inheritance. Red-green color blindness and hemophilia are examples of sex-linked inheritance in humans. The defective gene is located on X chromosome. Thus, a single defective gene causes disease in male, while two defective genes only can cause the disease is female. Female in heterozygous condition are apparently normal, but actually carriers of the disease. Female pass this defective gene to 50% of her sons. This disease is expressed only in males because male does not have the partner of the genes on Y chromosome. You can see this in figure. Try to figure out. Mitochondrial inheritance as a case of maternal inheritance. Apart from the nucleus, mitochondria and chloroplast also possess DNA, and you have just learned that genes are segments of DNA. Till now you have studied about genes on the chromosomes present in the nucleus. Since mitochondria come into the zygote from the egg, inheritance of mitochondrial DNA or DNA is said to be a case of maternal inheritance. In fact, certain diseases and therefore the genes responsible for them are due to defects in mitochondrial DNA and can be traced to family. Human karyotype. Human karyotype is the arrangement of chromosomes in 7 groups according to the types of chromosomes and their size. It is prepared by arranging chromosomes seen at mitotic metaphase. Total number of chromosomes 46. Number of autosomes 44. Sex chromosome X and Y is equal to 2. So depending on size, location of centromere and bands obtained by special staining method, human chromosomes are grouped into 7 groups A to G as shown in the figure. Sex determination in humans, as you have already learned, is as follows. Normal male 22 pairs of autosomes plus 1 X chromosome and 1 Y chromosome. Normal female 22 pairs of autosomes plus 2 X chromosome. Presence of Y is necessary for maleness. Absence of Y chromosome make the individual female. This is the figure called karyogram. Chromosomal abnormalities and genetic disorder in humans. Any change from the normal number of structure of chromosomes causes abnormalities. Following are some of them. Mongolism or Down syndrome. The individual has 47 chromosomes because of one extra chromosome in the 21st pair. This is trisomy. The outcome is that they are mentally retarded, have a thick tongue, and a dropping face. The possibility of giving birth to a Mongol child is far greater in pregnant mother above the age of 40. This is a diagram. So this is the Down syndrome. Trisomy 21 occurs when there are 3 copies of chromosomes 21 instead of the normal 2 copies. This disorder is also referred to as Down syndrome. About 5% of children born with Down syndrome have the familiar form. So the familiar form has a high risk of producing more than one. A Robertson translocation is a type of non-reciprocal translocation in which the long arm of two non-homologous ectocytic chromosomes become attached to from one chromosome with two long arms. The short arm attached together. These short arms carry non-essential genes and are usually lost in the cell after a few mitotic divisions. In this instance, a Robertson translocation occurred between chromosome 14 and 21. Heterozygous carriers of Robertson are phenotypically normal since they have two copies of each chromosome. However, the offspring of a normal parent and a carrier parent have an increased risk of being born with Down syndrome. The normal parent produces gametes having one copy of chromosome 14 and one of chromosome 21. The heterozygous parent produces 6 different gametes depending on how the chromosomes segregate during meiosis. The normal chromosome 14 and 21 may migrate to one pole while the translocated chromosome 14-21 migrate to the opposite pole. Upon fertilization, both zygotes are phenotypically normal. One zygote has two normal copies of chromosome 14 and 21 like the normal parent. The other zygote has a translocated chromosome 14-21 and a normal 14 and 21. So the translocated chromosome 14-21 may migrate to the same pole as the normal chromosome 21, leaving the normal chromosome 14 to migrate to the opposite pole. This case after fertilization, a zygote is formed with 3 copies of chromosome 21 and another zygote with only one copy. So the trisomic zygote will have Down syndrome and monosomic 21 zygote is inviable. So finally, the translocated chromosome 14-21 may migrate to the same pole as the normal chromosome 14, leaving chromosome 21 to migrate to the opposite pole. In this case, a zygote is produced, that is trisomy for chromosome 14 and the other is monosomic for chromosome 14. Both of these zygotes are inviable and do not survive. In short, one half of the zygote produced are inviable. Of those zygotes that survive to birth, one third are normal in chromosomal constitution. Klinefelter syndrome. Individual is a male with 47 chromosome with one extra X chromosome Typical features of a Klinefelter are tall, mentally retarded male, sterile and shows breast development. Turner syndrome, individual is a female with 45 chromosomes with only one X chromosome. The characteristic features are mentally retarded, web-like skin on neck, incompletely developed breasts. You can see in this figure. Color blindness and hemophilia. Both these are sex-linked disorders. The inheritance is as follows. You can see this in this figure. X is normal allele and XC is recessive mutant. Color blindness in male, the single X chromosome is received from the mother. Hence, a defective gene on X chromosome of the mother is passed on to the son and expressed as a defect. The daughter receives one X chromosome from the mother and the one from the father. The defective gene received from the mother may be masked by normal allele on the other X. The daughter is then called a carrier. Color-blind males are unable to distinguish between red-green colors. In hemophilia, affiliated male blood does not clot easily and the patient may bleed to death. Its mode of inheritance is exactly like thalassemia. It is a disorder in which hemoglobin is not synthesized properly, so frequent blood transfusions are required for survival. The defective gene is recessive and therefore heterozygous parents may not show the disorder. The child who gets the defective genes from both the parents suffer from thalassemia. Sickle cell. This is another hereditary abnormality due to mutation of a single gene in which RBC lost their shape and become sickle-shaped because of defective hemoglobin. Individuals possessing two defective genes cannot survive. In the heterozygous individual, one gene is normal, and so half the number of total RBC are normal, containing normal hemoglobin, while the others are defective. A boon in this gives against malaria for children with one defective hemoglobin gene can survive as they are less affected by malaria because the malaria parasite cannot thrive inside the defective RBCs. RS factor. RS factor is an antigen present on the surface of RBC. About 15% of all women do not have the gene for RH antigen, they are RH negative. Men can also be RH negative, but the problem which this trait creates is an RH negative woman. A pregnant RH negative woman whose husband is positive may bear a child who may have inherited the RH positive gene from the father. If the fetal blood enters mother's body stream, her immune system produces antibodies against RH antigen of the embryo. RH antigens cause no problem in first pregnancy, but in a subsequent early pregnancy, the mother's antibodies against RH antigen may enter the fetal blood stream and destroy its RBC, causing severe anemia, which may even be fatal. Nowadays, RS negative mother of RS positive fetus is treated immediately after delivery to destroy RH antigen in her bloodstream. Show the first pregnancy where the mother is RH negative and fetus is RH positive. Protein of the fetus stimulates the production of anti-factor in the mother, shows the retention of anti-factors in the mother's body, shows the RH positive fetus in the womb of the same mother during the second pregnancy, the anti-factors from the same mother during the second pregnancy. The anti-factors, this one repeat, will destroy the RBC. You can see this in diagram. This is the mechanism of RNA inheritance. Aminosynthesis. Aminosynthesis is a technique by which hereditary disorder due to defects in gene can be detected. In this technique, a small sample of amniotic fluid which surround the fetus is syringed out. This fluid has cells which break off from the skin of the fetus. Fetal cells are picked up and cultured. Chromosomes in the dividing cells are analyzed for genetic defects. If incurable genetic defects are detected, pregnancy can be terminated. It is illegal to use aminocentesis for detection of the unborn. The human genome. In the last over 100 years, genetics and molecular biology have gone far ahead, and the progress has been very rapid. You have just read about genetic disorder, and today there is hope for through gene therapy for hair care. This is because in 2003, most of the genes on human chromosome have been mapped or located on the 23rd chromosome. The gene responsible for inheritance of various structural features, genes that control various enzymes that catalyze the various biochemical reactions in the body, genes responsible for genetic disorder have been located. Genome means genes of a particular organism. Since genes are present in pairs, all kind of genes present in a particular type of organism are present on its haploid set. The human genome may be defined as all the genes present in the haploid set of chromosome in humans. There are an estimated 20,000 to 25,000 genes and 3 billion base pairs in the total human DNA. Each human chromosome has, apart from protein coding gene regulating base sequence, non-coding DNA promoter sequence in between genes that code for proteins. There are genes that code for the production of ribosomal RNA and the many transfer RNAs. It is estimated that only 1.5% of human genome has protein coding sequence and 20,000 to 25 genes. What you have learned? Heredity means the transmission of characters from parents to progeny. Variation pertains to difference between siblings or members of same species. Mendel was the first to explain that heredity involves transmission of units from reproductive cells of parents to offspring. Hugo de Vries, Currens and Sharma rediscovered the Mendel's law of inheritance. Mendel selected 7 varieties of garden pea, differing in 7 pairs of characters. According to his law of segregation, the factors segregate at the time of gamete formation. Mendel's law of dominance states that one factor dominates the other contrasting factor in phenotypic expression. Law of independent assortment states that the inheritance of one character does not depend on inheritance of any other character. There are deviations from Mendelian inheritance, and the pattern of inheritance are incomplete dominance, codominance, multiple allele, polygenetic inheritance, and pleiotropy. Sutton and Boveri proposed the chromosomal theory of heredity. It states that Mendelian factors or genes are located on chromosomes. Genes located on chromosome in a linear fashion and are held together in linkage group. Linked genes get segregated through chiasma formation or crossing over. Organisms with separate sexes have a pair of sex chromosomes called sex chromosome in human, X for female and XY for male. Male inherit an X chromosome from female parent and Y from the male. Y chromosome bears genes for maleness. Female receive 2 chromosomes, one each from either of the 2 parents. Any change from normal number and structure of chromosome causes abnormalities. A normal karyotype shows the 23 pairs of human chromosome and thousands of genes. Down syndrome patient have 47 chromosomes. This is trisomy of chromosome 21. Klinefelter syndrome patient has 44 autosomes and XXY. Turner syndrome has 44 autosomes, thus X0. Color blindness and hemophilia are X-linked disorder. Thalassemia and sickle cell anemia are due to a single defective gene. A positive fetus in a RS negative mother poses problem in which antibodies are produced in mother's blood against antigen of the fetus. The human genome has been mapped. Aminocentesis is a technique for detecting genetic